islands, the number of selves, let's just imagine this is a human embryo, the number of individuals in there is not fixed. It is not determined by the genetics. It is set in real time by the physiology. It could be 0, it could be 1, it could be 2, it could be some number of embryos, probably up to half a dozen, let's say. And it's very important for the making of selves that they are self-organized, meaning that they don't know, each cell does not know if every cell is some other cell's external environment. So where do I end and my neighbor begins, or where do I end and the outside world begins, right? This is something that is not true for almost any current AI or robotic system, because they don't have to put themselves together from scratch. They are told right from the beginning, they're programmed to say, this is your border, here are the inputs, here are the outputs, you are here, the outside world is there. That is not the case for living beings. We have to self-construct, and we do not know right away where our borders are, how many of us are in there, and so on. This also, taking it back to the brain, of course, is this issue of individuation, because in split brain patients and dissociative disorders and so on, you find out that there actually are more than one self within a standard human brain. How many could fit in there, we do not know. But the record for these dissociative kind of cases, I mean, I think I've read in the 60s that 60 different personalities that could be in there. So this idea of an active medium, an excitable medium that differentiates into a number of coherent selves is really key. And so now you have to start asking yourself, okay, so as we are self-constructing out of this sort of this Freudian ocean of possibility, some out of 20 or 50,000 cells, 3 or 2 or 1 embryo will form. What are the dynamics that actually allow individual cells with their own tiny little goals? What are the goals of cells? Well, every cell wants to become 2 cells, and they have metabolic goals that they try to meet and so on. What allows them to scale up to work towards very large, complex goals like building a whole embryo? I mean, that's huge. No individual cell can know what a whole embryo is, but the collective does. So what allows that? Well, here are some hypotheses. One thing that allows it, I think, is the kind of memory wiping that happens when cells are electrically connected. Because when 2 cells signal each other the old-fashioned way, where they just sort of secrete a chemical molecule and it comes over here and this cell senses it, it's very easy for this cell to know whether that signal came from the outside or not. So they can maintain, these 2 cells can maintain their individuality, and they can maintain different memories. And this cell can ignore the signal or do something about it, whatever. But there's something magic about these electrical gap junctions. When the cells are joined by these gap junctions, this signal, let's say it's a calcium spike from being injured. This cell got poked with something, and this calcium spike is a trace of that event, a kind of a memory engram of that event. It will immediately propagate through these gap junctions, and the interpretation of that signal for this cell is exactly the same as it would be for its own calcium spike. Calcium doesn't have any metadata on it that says, where did you come from? So by moving through this, there's a kind of memory, kind of identity wipe that happens, where this cell now has that same memory trace, and as far as it's concerned, that's its own natural memory. It has no way of knowing that's a false memory. So to this cell, that's a false memory, but to the collective, it is an absolutely true memory that the collective had. So that kind of wiping property, it strips ownership information. It makes it very hard for each cell to maintain an individual identity with respect to what its history was, and it makes collectives out of individual cells. And of course, it makes them bigger. So my memory become our memories, my stress state becomes our stress, and my goal becomes our goal. And so when a single cell in this diagram, when a single cell has some size of a cognitive light cone and some ability to anticipate and remember, a network of these cells has a bigger computational capacity and a bigger spatial sensorium, and it can actually have larger scale, more complex states as set points. And now you can start to see how a collection of cells could actually work on something like building an arm or building a whole body. And so we can imagine, and so we're doing this now, is doing these very specific computational models of how little tiny homeostatic loops like this, for example, measure the pH and ACT, become very large homeostatic loops about something completely different. Single cells cannot remember anything like this, but a giant collection of cells could. And so this is a very active area of research, is to figure out exactly how that scales up. The implications are these. Here's what evolution has done. Evolution has enabled single cells to work together to enlarge their cognitive light cones, where the goals scale from the very humble goals of single cells to the more grandiose goals of collectives, where they can work on something gigantic, like making sure you have 4 fingers. This cell doesn't know anything about what a finger is, but this collective does. So evolution gave us this scale up, but there's a failure mode to this. The failure mode is cancer. So here's where when these cells are electrically isolated from each other, this is human glioblastoma in culture. So these individual cells can roll back to their unicellular past. They do not become more selfish. A lot of game theory kind of approaches treat cancer cells as more selfish. They don't become more selfish. Their selves get smaller. They're just as selfish. It's just that the self here is huge. Their self is very tiny, and they do the best they can. But biomedically, if you have that kind of weird perspective on cancer as a disorder of selfhood, then what you can do is something interesting. And we did this years ago. You can induce tumors using human oncogenes, but what you can do is use these ion channels to force the cells to be in the correct electrical state to maintain proper communication. And when you do that, even though the oncogene is blazingly expressed, it's labeled in red here, you can see it's all over the place. There's no tumor. This is the same animal. There's no tumor because the genetics might have a defect. The hardware might be a little bit broken, but as long as the cell is physiologically part of this network that contains large-scale goals like building nice skin, nice muscle, and so on, they're kept away from tumorigenesis and harnessed towards these large-scale anatomical goals. Okay, so the last piece I want to show you, it goes beyond this. So we've been talking about goals in different spaces. So the tiny metabolic goals of single cells, the morphogenetic goals of cellular collectives, and now we can ask the following question. Where do these goals come from, and what other goals can novel beings have? And so this is a project I also have to do a disclosure here because Josh Bongard and I have another company called Fauna Systems, which is all about AI designed biorobotics. And this is all work done in collaboration with the Bongard Lab at this Institute of Computationally Designed Organisms. We asked the following question. How much plasticity do these cells have? And when they're liberated from their normal boundary conditions of the animal, what will they do? How will they reboot multicellularity? How will they navigate anatomical and behavioral spaces? What can they do? So here's the